Hello, future pharmacists and science enthusiasts. Today we're diving into a topic that is fundamental to making medicines. It's called size separation. Imagine you have sand and pebbles mixed together. Separating them by size is exactly what we do in pharmaceuticals. But instead of sand, we work with powders that become the medicines we rely on. Particle size dramatically affects how drugs behave in the body. Smaller particles dissolve faster, leading to quicker absorption and faster relief. Uniform particle size ensures every dose is consistent. Size separation is a cornerstone of pharmaceutical quality control, ensuring safe and effective medicines. Think about baking a cake. Lumpy flour creates uneven results. The same principle applies to medicines. We need uniform mixing for consistent results. Different sized particles separate during processing, leading to dose variations. Consistent particle size creates the perfect medicinal blend. By controlling particle size, we control drug performance and patient safety. While it might sound like simple sorting, size separation directly impacts health and well-being. Science is cool, isn't it? Our main goal is improving drug bioavailability, how well medicine gets absorbed by the body. Smaller particles dissolve faster, crucial for emergency medications. Another key objective is dose uniformity. Each tablet must contain exactly the right amount. Without uniform particle size, some tablets get too much drug, others too little. Size separation ensures physical stability in liquid medicines like children's antibiotics. Large particles settle quickly, making the last doses ineffective. Fine, uniform particles create stable suspensions with consistent dosing. Size separation applications are everywhere in pharmaceutical manufacturing, essential for tablet production, critical for capsule filling, paramount for inhalers, where particles must be small enough to reach deep into lungs. From quality control labs testing raw materials, to final production stages, size separation is an indispensable tool for the modern pharmaceutical scientist, ensuring every medicine meets the highest standards of quality and effectiveness. The most common method is sieving, beautifully simple, like using a colander to drain pasta. Smaller particles fall through the mesh, larger particles are retained on top. But particles must be in motion to find the openings. Static powder won't separate effectively. Vibration makes particles bounce and present to the mesh, breaking up clumps. Gyration moves the sieve in circular motion, spreading powder gently across the surface. Brushing continuously sweeps the surface and clears stuck particles from apertures. Advanced shakers combine multiple motions for optimal separation efficiency. This creates direct mechanical sorting into distinct size fractions. All agitation methods aim to move particles so they can be challenged by the sieve openings. Whether shaking, swirling, or brushing, the goal is efficient, reproducible separation. The result is precise particle size distribution data that pharmaceutical scientists use to ensure product quality and performance. This mechanical approach provides reliable, quantitative results that meet regulatory standards. In medicine, we need universal standards. Official pharmacopoeias provide specific standards for powders and sieves. United States Pharmacopoeia, British Pharmacopoeia, these create a universal language for particle size. These standards are not suggestions, they are mandatory rules for global drug manufacturing. Powders are classified into grades: very coarse, coarse, moderately coarse, fine, and very fine. Each grade is defined by specific sieve numbers that particles must pass through. For example, very coarse powder passes through sieve number 8, while very fine powder passes through sieve number 120. Official sieves are precision instruments, not simple kitchen strainers. Standards specify everything: wire diameter, aperture size, and permissible variations. Sieve numbers indicate openings per linear inch. Higher numbers mean smaller openings. Constructed from non-reactive materials like stainless steel mesh on brass frames, these precision instruments require careful handling and regular calibration. By using standardized sieves to classify standardized powder grades, we build a system of quality and consistency that underpins the entire pharmaceutical industry, ensuring medicines are safe and effective worldwide. Sieving is based on particle dimensions relative to sieve apertures, but the process is more complex than it appears. Particle orientation is crucial. A needle-shaped particle might be too long when flat, but can slip through end first. This is why agitation is essential. It gives particles multiple chances for favorable orientation. Sieving duration is critical. Brief shaking leaves many particles without time to find openings. Sieving continues until an endpoint when less than 0.1% of sample weight passes per minute. Overloading creates deep beds where particles can't reach the mesh efficiently. Each sieve diameter has an optimal sample size for accurate results. Powder physical properties greatly influence sieving success. Sticky or electrostatically charged particles clump together, acting like larger particles. Moist or hygroscopic powders are difficult to sieve and may require flow aids. Controlling humidity and adding flow aids like magnesium stearate can improve sieving. Understanding these principles, orientation, time, loading, and material properties, separates a technician from a true pharmaceutical scientist. Mastering these factors ensures accurate, reproducible particle size analysis. Shaking sieves by hand for 15 minutes is tedious and not reproducible. The mechanical sieve shaker eliminates human error and effort, providing consistent agitation. A sieve shaker has a motor-driven base, platform, clamping mechanism, sieves, lid, and receiving pan. Sieves are stacked with coarsest at top, descending to finest, with a solid pan at bottom. The row-tap shaker combines horizontal circular motion with vertical tapping action. First, weigh your sample accurately. Assemble clean sieves in descending aperture size. Pour sample onto top sieve and secure with lid. Clamp in shaker, set timer for 10 minutes and start the machine. After completion, disassemble and weigh powder retained on each sieve. Circular motion spreads powder across the entire sieve surface. Vertical tapping jolts particles through openings and prevents mesh blinding. This combination provides reproducible, standardized particle size analysis. The mechanical sieve shaker transforms a manual process into precise scientific measurement, essential for pharmaceutical quality control. Sieve shakers offer simplicity and affordability compared to advanced techniques like laser diffraction. Easy to understand and perform, ideal for routine QC with minimal training required. Mechanical shakers eliminate manual variability, providing the same motion for the same time, every time. Provides direct, physical measurement. You can literally see the separated fractions. Process is time-consuming, typically requiring 10 to 20 minutes plus preparation and cleanup. For particles smaller than 45 micrometers, sieving becomes inefficient and inaccurate. Sieving is dusty and noisy, sometimes requiring protective equipment and sound enclosures. Aggressive agitation can break fragile particles, skewing results toward finer sizes. Primary use is particle size distribution of raw materials, APIs, and excipients, critical for monitoring in-process granules and ensuring materials meet specifications. Essential for determining compliance with official pharmacopoeial powder grades. In R&D, used to study milling effects and develop optimal formulations for flow and compaction. Despite limitations, sieve shakers remain indispensable for ensuring quality from raw materials to finished solid dosage forms, supporting the pharmaceutical industry's commitment to safe, effective medicines.